Hello dear friends and my dear students. Today I am going to start with a new chapter, which is chapter number 10, Human Health and Diseases part 1. In this part we are going to study about health, our immune system, antigens, antibodies, their complexes, and about the blood group, that is ABO blood group in human. So let's get started with health. According to World Health Organization, health is defined as the state of complete physical, mental and social well-being of a person. If a person is physically, mentally and socially well, that time a person is called a healthy person, not merely the absence of diseases. So to achieve a good health, it is always necessary that we have hygienic balanced diet, clean drinking water, personal and community hygiene, regular physical exercise, knowledge about the diseases and their effect in our body, the proper disposal of waste and the control of vectors. Now what is immune system? As we all know that always our body is exposed to so many of foreign particles or viruses or bacteria. So despite of constant exposure of the variety of pathogen, most of us remain healthy. So what is the reason behind that? So this is the fact that the human body has ability to resist almost all type of those foreign bodies. Then the system which protects us from various infectious agent is called our immune system. So that immune system which is there inside our body, which always protect us from different type of infectious agents. So resistance is the ability to prevent the damage or disease through our defense mechanism, which is our immune system. So the immune system have a capacity to differentiate between the body cells, which are our own cells, and some invaders, which are foreign particles or foreign pathogens. They may be bacteria or any microorganisms. So any foreign substance invading body and capable of stimulating an immune response, it is called an antigen. As soon as foreign body or some pathogen invade our body, they will immune response will be stimulated, and that is called an antigen. And some protective chemical will be produced by immune cells in response to those antigen. They are called as antibodies. Okay, so did you get my point? Antigens are the foreign particle which are infected our body, and the antibodies which are the chemicals they produced against those antigens. Now next is immunity. The concept of immunity is believed to be started by Edward Jenner in England. Immunity is referred as a resistance exhibit by the host toward the injury caused by pathogen and their products. So there are two different kinds of immunity. One is innate, or that is also known as inborn or inherited immunity, which is since the childhood, childbirth. And acquired is also known as adaptive immunity, which is acquired in the further life. So first is innate or inherited or inborn immunity. It is the natural or inborn defense system of the body. It is not affected by prior contact with microorganism or immunization. So one form of innate immunity comprises various types of barrier which prevent entry of foreign agents into the body. So which is there since your childhood into the person. And the acquired immunity, the resistance that an individual acquires during their life. Okay, so it is acquired immunity. It is also known as adaptive or some specific immunity. This involves the formation of antibodies in the body which neutralize those antigens which are affecting their bodies. So acquired or adaptive immunity has the following unique features. So one is specificity. Specificity means it can produce a specific antibody against a particular antigen. Okay, so specificity means the by word itself we can identify that they are specific for some antibodies against some specific antigen. Then number 2 is diversity. It can recognize a vast variety of diverse pathogen or some foreign molecules. So diversity means they can recognize or identify many kind of pathogens. Number 3 is discrimination between self and non-self. Okay, this is again a important feature, that is it differentiate between body cell like own body cells and some foreign molecules, they are non-self. And number 4 is memory. Memory, when the immune system encounters a specific kind of foreign agent for the first time, it generates an immune response and eliminate the invader. This is called first encounter. So once it will identify some foreign agent first time, it will generate an immune system and it will remain into its memory for further. So same kind of foreign antigen or foreign particle will invade the body again, they would be able to identify. That is why this is the feature, that memory is the feature, unique feature of acquired immunity. So these kind of those 4 features are important features of acquired immunity. Now types of acquired immunity. Acquired immunity is of 2 types, one is active and another one is passive one. Okay, so active immunity, it is resistance developed by individual as a result of an antigenic stimulus and artificial, it may be natural or it may be sometime artificial also. So natural is for lifetime and artificial is only for, it may be temporary or it may be permanent type. Then second one is passive immunity. Passive immunity is also known as its passive immunity is acquired when ready-made antibodies are received by body cell, that is body cell do not take any active part in the production. As the word name itself you can identify that they are not taking active part in the production of immunity. It also can be natural as well as artificial. So let's take active immunity. Active immunity, they are of 2 types, one is natural acquired active immunity and second one is artificial acquired active immunity. So what is natural acquired active immunity? Is immunity acquired due to infection is called natural acquired immunity, active immunity. And artificial acquired, it is the resistance induced by vaccines. So vaccine is introduced into the body to stimulate the formation of antibodies by the immune system. Example are polio vaccines or BCG vaccines, we know very well. Then what is passive immunity? Passive immunity is again of 2 types, one is natural and second one is artificial. So passive, natural acquired passive immunity. So before birth, maternal antibodies are transferred from the mother to the fetus through the placenta, and after birth antibodies are transferred from the mother to the infant through the colostrum, which is first milk from the mother. Then artificial acquired passive immunity, this immunity is developed by injecting previously prepared antibodies using serum from the human or the animals. So this is about artificial acquired passive immunity. So these are the 2 important type of acquired immunity. Now cells for immune system, this is really important, that there are 2 main types of cells are involved in the working of the immune system. One is lymphocytes and second one is antigen presenting cells, they are also known as APCs. So what is lymphocyte? Lymphocytes are the main cells of immune system. They are like the other blood corpuscles, arises from the stem cells present in the liver of fetus and in the bone marrow in the adults. What is bone marrow? Bone marrow is actually a soft tissue which are present specifically in the thighs or the hips tissues. So some of them, they undergo differentiation in the gut associated basal lymphoid tissue are called B lymphocytes, and other are differentiated in the thymus gland and they are termed as a T lymphocytes. So there are 2 different lymphocytes are present, B lymphocytes and T lymphocytes. So the mature lymphocytes, they passes into body fluid, that is blood and lymph. Lymph is a transparent fluid which circulates in the lymphatic system of the person. So the mature lymphocytes passes into the body fluids and they circulate into the body. The B lymphocytes form antibody mediated immune system, that is AMIS, and T lymphocytes, they form cell mediated immune system, that is CMIS. So both the immune system, they need antigens to come into action, but they respond in different ways. So AMIS and CMIS, they both the system, they need antigens to come into action, but the response are different. Then next is mechanism of response of T lymphocytes to the antigen. T lymphocytes form clones of T cells, and when it come into the contact with an antigen, they are similar but perform different functions. The clone has 4 different type of lymphocytes. So let's have one by one. One is helper T cell. By name itself you can identify helper T cells. So they produce lymphokines for performing functions like proliferation of other T cell, stimulation of B lymphocytes and macrophages. So they are always there to help in performing some functions. Then number 2 is killer T cells. Killer T cells, also known as cytotoxic T cells. So killer T cells bind to infected cell and secrete perforins. Perforins, these are the proteins which are required, or they infect the cell and they make hole into cell and they kill those cells. So those perforins, they form a hole in the infected cells, and it also releases substances that kills the cell. So the name is cytotoxic. Cytotoxic means they are killer cells. Okay, so they directly attack and destroy the cancer cells also. Then third one is suppressor T cells. Suppressor T cells, these cells suppresses the entire immune system against the attack on the own body cells. Then fourth one is memory T cells. So these are previously sanitized cells which retain the sanitization memory for the long time in the future. So that is how there are 4 different type of cells: helper cells, killer T cells, suppressor cell and the memory T cells. Now what is the mechanism of action of B lymphocytes to antigens? So activated B lymphocytes multiply very fast to produce clones of plasma cells and memory B cells. The plasma cells produces specialized glycoprotein which are called antibodies, which are circulated through the body fluid like blood and lymph. We have already studied. The free antibodies, they have 3 main functions. One is agglutination. Agglutination means it is a clumping of particles. Okay, so agglutination, that is clumping of particulate matter which includes some bacteria or viruses, and then they engulf by the phagocytes. Okay, phagocytes means the engulfing cells. Then second one is opsonization, which is also known as coating. So coating of bacteria to facilitate their subsequent phagocytosis by the macrophages. And third one is neutralization of toxins released by the bacteria. Example is tetanus toxin. So important point here is that each antibody is always specific for a particular antigen. That is how they form antigen antibody complex. Then next is about antigen presenting cells. So we have already studied about the lymphocyte, T lymphocyte and B lymphocyte, and the second part is about APCs, which is antigen presenting cells. So antigen presenting cell engulf and invading pathogens and process the antigens. Then the processed antigen are presented on their own surface. These cells are able to deliver a stimulatory signal that is necessary for the activation of the T cell. Okay. Then what is vaccination? So administration of any vaccine to protect against any further pathogen, virus, bacteria. So the administration of vaccine to protect against a particular pathogen is called a vaccination. The body's immune system helps to protect against pathogen that cause infection. The vaccination is always an important form of primary prevention, which allow us to control diseases like measles, polio, tetanus and whooping cough that once threatened many lives. Then what is the structure of the antibody? Antibodies are the glycoproteins. These are the glycoproteins which are highly specific to a specific antigens. They are also known as immunoglobulins, which is denoted by IgS, and produced in response to antigenic stimulation. The antibodies are produced by the plasma cells. This is important that these antibodies, they are produced by what? Plasma cell, which in turn are formed by B lymphocytes. The mature plasma cells produce antibodies at an extremely rapid rate, that is about 2000 molecules per second. So that is how those antibodies are produced into the body of a human. So this is the structure of antibody, where you can say this is a Y-shaped molecule. So antibody is always a Y-shaped molecule, and each immunoglobulin molecule is made up of 4 polypeptide chains. So you can see there are 4 polypeptide chains are present, and there are 2 heavy chains which are known as H chains. So 2 heavy chains are present and 2 light chains are present, which are denoted by L. Okay, so you can see in the diagram that those brown are the heavy chains and those blue one are the light chains. So blue, heavy and light chains also, they have different shades. So the upper one, the darker one, is a variable region of the heavy chain. So darker brown one is a variable region, and lighter brown one is a constant region of the heavy chain, rather than the blue one where it is in a light blue. Light blue is a constant region of the light chain, and the dark blue is a variable region of the light chain. Okay, so the 4 polypeptide chains are held together by those disulfide bond to form a Y-shaped structure. The result holding together arms and stem of the body is termed as a hinge joints. So each chain of the antibody that include 2 distinct regions, so variable region and the antigen binding site, it is paratop. So this part of antibody recognize the bind of a specific antigen to form a antibody complex. Okay, so you will find those darker side of both the chains, they are having NN, those are known as antigen binding sites. So that is how they form a antigen antibody complex. So this is the structure of a antibody, which is a Y-shaped molecule. Next is formation of antigen antibody complex. The study of antigen antibody interaction is called serology. So each antibody is specific for a particular antigen, we have already studied. Combining sites of antigen is called antigenic determinant, that is also known as epitopes. They react with the corresponding antigen binding site of antibody, that is known as paratop. So on the specific site on antigen is known as epitopes, and a specific site on the antibody, that is known as paratops. The antigen binding site, which is paratops, are located on the variable region of the antibody. In the structure of antibody, we just have studied about the paratops. So small variation in the variable region makes each antibody highly specific for a particular antigen. The variable region enables the antibody to recognize a specific antigen and bind to specific antigen in a lock and key forming and they form a antigen antibody complex. So we have studied in 11th standard that the antigen antibody complex form, which is a lock and key model. So specific antigen will specific bind to the a specific antibody. So here is a diagram. In this diagram you can see this is a virus, which is a hexagonal structure of a virus, and where small circular structure which are the antigen determinants, also known as epitopes. And here in this diagram you will find those purple and red color of antibodies are present, and those specific antibody, they have paratopes of antibodies bound on the epitope. So specific kind of antigen will bind on the specific kind of antibody only. And our next is ABO blood groups. So the A, B and O blood groups were discovered by Karl Landsteiner in 1900. Landsteiner was awarded with the Nobel Prize for his discovery of human blood groups. Okay, so he found two antigen and agglutinias on the surface of human red cells, and they are named as antigen A and antigen B. So in ABO system of blood, the blood groups are determined by the presence or the absence of antigen A and the antigen B. So here is a ABO blood group in human. So I will just brief you about blood group A. So what is blood group A? When an individual with blood group A have the antigen A on the surface of their red blood cells and the antibody B in their plasma. What is blood group B? The blood group B individual with blood group B have the antigen B on the surface of their RBCs and antibodies A in their plasma. Then what is blood group AB? If individual with blood group AB have both antigens A and B on their surface of RBC and no antibodies in their plasma. Then blood group O is individual with blood group O lack both the antigens A and B on the surface of their RBCs, and this shows the presence of both A and B antibodies in their plasma. So in this diagram you can see that the blood group A is having genotype is IAA, IAIA or IAIO also. The antigen on the surface of RBC will be capital A. The antibody in the serum will be small antibody B. Then they can donate blood to A, blood group A, and also they can donate blood to the AB blood system. They can receive blood from A and O always, because O is always a universal donor. Likewise, you can see in the B, the genotype for B blood group is IB or IBIO. The antigen on the surface of RBC is B, capital B. Antibody in the serum is antibody small a, can donate blood to B, and AB can receive blood from B and O. What is about AB blood group, which is always known as universal acceptor. So their genotype is IA and IB. Antigen on the surface is capital A, capital B. Antibody on the serum is nil, as I have already told you. They can donate blood to only AB system, and they can receive blood from A, B, AB and O. All four can donate blood to the AB, as they are universal acceptor. Then next is blood group O, which is always known as universal donor. So their genotype is IOIO. Antigen on surface is nil. Antibody in the serum is both antibody A and antibody B. Can donate blood to A, B, AB and O, and can only receive blood from O because it is universal donor. So this is all about the immune system, our blood group system, antigens, antibodies, and in my next video I will explain you about common human diseases. So thank you